Hi, everyone. Thank you so much for taking the time to come today. I actually don't know a lot of you, so let me introduce myself. My name is Sallie Schneider, and I'm Director of the Histology and Tissue Procurement Facility, also known as HistoSpring, which resides down at the Pioneer Valley Life Sciences Institute. And we're very excited actually to talk about some new capabilities that we have, brought to you by some exciting stuff from advanced cell diagnostics and some new equipment that we have. And so we're thrilled to be able to come in and share this with you. We will have two speakers today. We'll have Dr. Blake McAlpin from Advanced Cell Diagnostics, followed by Dr. Kelly Gregory from HistoSpring. And they will be talking about their various portions of this, and at the very end, we'll have a question and answer session. So just a little housekeeping. We're going to keep all the questions till the end, if that's okay. We will take questions from virtual participants as well. Just for those of you out there, just submit through the chat, please. All the in-person attendees can ask questions as they would like. This is being recorded. I want to let you know that. And there will be access to the slides and the recording in a couple of days if you sign up for this, okay? All right, so this is just to say that we are going to go through some advanced histology and spatial biology at HistoSpring. We were able to get a Mass Life Science grant recently, and with that, got some new capital equipment. We got the Bond RXM, cell scanner, and some Halo software. So Kelly's going to be talking about all that. So with that, I'm going to introduce our first speaker, Dr. Blake McAlpin. He received his bachelor's degree in neuroscience from the University of Michigan. His master's degree in Paris from the University of focused on endocrine disruption by BPA with regard to endogenous circadian clocks. And he got his Ph.D. in neuroscience from the MD Anderson University of Texas Health Graduate School of Biomedical Sciences in 2021, where he studied neurotoxic side effects of chemotherapy treatment with a focus of microglia-mediated mechanisms underlying cognitive dysfunction. So Blake is now currently a field application scientist in the Boston area for advanced cell diagnostics, and we are very grateful that he was able to come out today and talk to us about this RNAscope. So Blake, thank you.
Thank you. That made me sound very good, so thank you for all that. My name is Blake McAlpin. I'm a field application scientist for ACD. ACD is the company that makes RNAscope. So today I'm going to kind of just go over the technology, go over some examples of how it's been used, and feel free to stop me at any time in the talk. This is kind of an informal thing, so if you have questions, you can raise your hand and I will get to you. So the title of this talk is Advancing Spatial Biology at HistoSpring, which is associated with UMass Amherst, same section RNA and protein detection with RNAscope. Do I need this on or no? Okay.
I think maybe for
hearing you.
Zoom, maybe for Zoom, you want to double check that.
This should work if he's standing in front of the thing.
I was hearing it. I'll check again.
Okay.
Keep talking.
All right. Okay, so today I'm just going to go over RNAscope technology overview, kind of talk about how it works. Does anybody in this room currently use RNAscope? Okay, nice. And does it work okay? Okay, great. Good. So yeah, we'll go over RNAscope technology to people who don't know it. Then we'll go over some examples of how it's been used. So just show you some spatial mapping of transcriptomic data. Then we'll talk about combining RNAscope with protein co-detection, because that's something a lot of people do nowadays. And then we'll introduce to you our newest assay, which is called the multi-omic assay. And this is a RNA protein combination assay. So what is RNAscope? Who are we? So ACD is Advanced Cell Diagnostics, and we created RNAscope, which is essentially in situ hybridization. So it's a way to look at RNA transcripts in a spatial realm. RNAscope is very popular, so it's been used in over 10,000 publications. And out of those publications, 25% actually do RNA plus protein combined. We're not a huge company. We've got like 200 people. This is all to say that we've been around for 10 years and it's kind of an accepted technology in biomedical research. And it's also used in the clinical side as well to diagnose certain things. RNAscope, we have 50,000 probes that have been designed. So a probe is what we call the little thing that targets your genes. But the important thing to note is that if we do not have a probe designed to target your transcript of interest, we can design one for you. So we have a team of Ph.D. scientists in our probe design group. You would basically submit your sequence and we can actually just design a probe, run it through an algorithm, design a probe that will target that sequence without cross detection of, let's say, like your endogenous genome. In addition to that, all of our probes, they all have the same melting temperature. So back in the day when you used to have to optimize ISH, your incubation could be for 2 hours, it could be for 4 days. So there was a lot of variability in the melting temperature for your oligonucleotides. With RNAscope, they all melt at 40 degrees Celsius after 2 hours. So that's a really big advantage. You don't have to worry about optimizing anything after the hybridization point. Okay, so let's just get into kind of the workflow. So if no one has done, if you haven't done RNAscope, I like to compare it to like immunofluorescence or IHC because it's kind of a similar concept. The workflow is after the hybridization point here, the workflow is standard. So you would not really adjust pretty much anything aside from maybe fluorophore concentration. But this first step, this permeabilization step, this is the most important part of the assay. Essentially, you need to permeabilize your tissue in order for the probes to penetrate and hybridize to your transcript. So this permeabilization step, we use a target retrieval or an antigen retrieval to reverse fixation. And then we have either a protease or a protease-free reagent to permeabilize the membrane. And I'll go over that later because this protease-free thing is kind of new. And that's what a lot of people use when they try to combine with an antibody. So this needs to be optimized depending on your tissue. But after that, you just follow the protocol. Everything should be kind of turnkey. We have a hybridization step, which again is the same for every probe, every gene target. And then there's a series of amplification incubations. So this is not actually amplifying your RNA. That's not PCR. We're actually building a structure on top of the probe because it's such a tiny thing that if you just added a fluorophore to that probe, you wouldn't see anything. It would be invisible to the eye, even under a microscope. So we have to build this amplification tree, and then you'll have a lot of sites available for a chromogen or a fluorophore to bind. And then you can actually see them just with a normal microscope. You don't need any like fancy detection capabilities. Any questions about that? Sorry, it's a little dense. Okay. So this is what a chromogenic result looks like. So this is a relatively highly expressed gene target. One thing that's important to note, and I'll use your little guy here, is that each one of these dots is one RNA transcript. You'll notice that some dots are larger and some dots are smaller. That still represents one transcript. The reason one dot might be larger is, let's say you have 100% of your RNA is preserved in this area of the tissue. And this smaller dot was in the process of being degraded because RNA is very labile. It doesn't stick around for what it's supposed to degrade. So even though you might have a small dot versus a large dot, we still consider that one transcript. So when you quantify RNAscope, the best way to do it is to count the dots. So when I talk about an RNAscope probe, this is what I mean. In the past, in situ hybridization, you'd have a very long oligo sequence that would have to hybridize on the entire transcript. The way RNAscope probes are designed, in one vial of probe, it's not actually just one long sequence. It's a bunch of these little chopped up sequences that we call ZZ pairs. And each ZZ pair is 50 base pairs long. So if you're targeting an 1,000 nucleotide sequence, you actually have 20 of these ZZ pairs mixed together in a liquid. And the benefit to that is, one, you can design probes to be very specific because you don't need to, target this huge region. You can just target small regions within the gene that are unique to that gene. And the other really important thing is that, like I just said, you have RNA that degrades all the time. So you're never going to have perfect RNA quality. So even if you have like 50% of your RNA is degraded, really you only need 3 to 6 of these easy pairs out of 20 to hybridize to your transcript in order to see signal. So that's why I was saying a large dot, maybe you have 20 of these easy pairs on your transcript versus a small dot, you have 3, but that still is the presence of a transcript. Any questions about that? Awesome. So when we run the assay, this is kind of what it looks like in the tissue. So you have your target sequence. This is the RNA that you're trying to look at. And then you'll have your probes hybridized directly to your transcript. So you've got around 20 of these easy pairs that are now hybridized to your transcript, and we're just looking at one. Then there's the series of amplification steps I talked about. So you just incubate a reagent, AMP1, AMP2, AMP3, and what you're doing is actually building this little amplification tree. And then what you do at the end is you have all these binding sites that are available. You either add a chromogen or a fluorophore. You can do either one. And why the double Z? It's for this reason. RNA is very homologous. There's a lot of repeats. So if one Z binds off target, which it's going to do because RNA has a lot of repeats, that's totally fine because you need both of them to hybridize directly next to each other in order for this preamplifier molecule to bind. So in that sense, we have not only sensitivity, which means you can detect very, very small things, but we have specificity too. And there are some RNAscope assays where you can combine multiple RNA targets. Just briefly, how do we do that? Well, this top sequence, this is an oligonucleotide sequence. This will be unique to your C1 probe, and then your C2 probe will have a totally different sequence. So when you use that AMP1 reagent, that preamplifier molecule, it's actually a mixture of all of the different probe channels preamplifier molecules. So you'll have C1 through, you can even go to C6 mixed together. All of these molecules, the C1 through C6 preamplifier will also be in the reagent. So you can amplify all of them at the same time without, cross detection. So this is kind of just all of the different types of assays that we offer. So it's not just one RNAscope assay, there's different assays depending on the length of RNA you'd like to target. So we have assays that target microRNA targets from 17 to 50 nucleotides, all the way up to full-length RNAs, 1,000 nucleotides. In the middle, we have this base scope assay, which is like, instead of having 20 ZZ pairs in that solution, you have 1 to 3. So the difference between the RNAscope and base scope is that you have more amplification steps because you're targeting a smaller region. And why would you want to target a smaller region? Let's say you want to target like a specific exon-exon junction. You can put one Z here, you can put one Z here, and then if they're, if the exon-exon junction meets, then you'll see signal. I'm not really going to go into too much detail. If you want, you can take a picture of this. The recording will also be sent to you, so you can go back. This is a dense slide. I'm also not going to go over it. This is just everything that we offer. These are all of the different assays we have, from chromogenic to fluorescent, from single plex all the way up to 12 or 48 plex. So again, I'm not going to go over this just because it's dense. You can take a picture of it if you want, or you could just look at the recording. But if you have any questions about our assays, you can ask me, you can ask Hank. Hank is your account executive, and then we can work with you to get it set up. And if you're interested in automating it, you should talk to Kelly. Speaking of automation, that was perfect. Speaking of automation, Kelly has a Leica bond. So a Leica bond, I didn't hear about this until I got out of academia, because everyone just does it themselves because we're cheaper than a Leica bond, PhD students. But the Leica bond is an auto stainer. So basically, it's this machine where you can stick your slides on it, and it has all of these different reagents that you can stick into the machine, and you can program it and you can tell it when to add or remove different reagents. So our RNAscope assays also run on these machines. You can run like 30 slides overnight, depending on the bandwidth. So that's just to introduce you to this concept that RNAscope can run on this, and Kelly has one.
So quick question, just
Yeah.
to clarify, that's a separate kit for the Lycobot.
It's
So
different.
if you normally have all your probes and everything else, it would be separate.
Yeah, so the probe design itself is the exact same. It's just that it's diluted in a different buffer. So you would actually have to purchase a new probe, yes. But it would be the exact same like readout that you've done in the past. But yeah. Because it does run through more reagents, so the little dropper bottles, yeah. Okay, so now we're gonna go over some RNAscope data just to give you some ideas of how it's been used in the past and how you could use it. So this one is kind of the most basic use for RNAscope. So let's say you're having trouble with an antibody stain. A lot of times you might, I think I just read a paper actually where they had like 12 different antibodies and only 2 consistently targeted what it said it was actually targeting. So there's a lot of variability in antibodies depending on who you buy it from, if it's monoclonal or polyclonal. So if you're able to do your readout with RNA, you don't necessarily need the protein. RNAscope is a great solution for no antibodies or for validating an antibody if you want to look at co-localization. So in this example, there were researchers who were trying to look at LGR5 and they noticed that depending on where they got their antibody, they would get different results. So then they decided to use RNAscope. And we've got this little quote that says, using RNAscope assay probe against LGR5 RNA has allowed us to study the localization of LGR5 expression without relying on an antibody. So the probes are just oligo sequences. So we make these probes, they're always the same. We're not taking them out from an animal or something. So it's just a sequence. It's like ordering a primer. There's never variability in the probes. Not like significant variability. I heard that there was some interest in microRNA targets. So here's an example of an assay we have which targets 1 microRNA or small RNA target combined with 3 full-length RNA targets. So these are housekeeping genes. This is just an example of what it looks like. You've got your PPIB. It doesn't look very great, to be honest, on this screen. But maybe a little better over here. You have these little puncta. So all of these little dots are RNAscope transcripts. It usually looks a little brighter, but you can still kind of see PPIB, UBC, and POLR2A. Those are housekeeping genes. And then you've got your microRNA probe. So this is what the readout looks like. This is a highly expressed endogenous microRNA. This can also be automated on the Lycobot. Here's an example of co-detection. So in this experiment, researchers wanted to label the protein. So they labeled CD8. And what you can do, let's say you have, like interferon gamma is kind of difficult to target with an antibody. You can use RNAscope to target it. And then you can look at co-localization with your protein and your RNA to identify cell types or like activation types states. So you've got cytotoxic lymphocytes when CD8 and interferon gamma co-localized, and then you also have Tregs when CD4, which is here is RNA target green, and FOXP3, which is kind of orange when they co-localize. Okay, so I'm not sure if there's any gene therapy research being done, but this kind of gives you an understanding of how you can use our probe design in like maybe a not intuitive way in order to look at different things. So a normal RNAscope probe targets RNA. It's antisense, so it targets RNA, and technically it would also target episomal DNA, which really shouldn't exist in your tissues unless you've introduced a vector. So it can target both of those things because the DNA denatures. However, if you're only interested in targeting DNA, you can design a probe that's like inverse from a normal RNAscope probe, and then it's only going to detect the DNA strand. So what I'm showing here is that researchers often use, like, let's say they design a sense probe, and you could look at a promoter sequence. So you're introducing a gene into an animal to fix some disease. So you'll introduce this vector, and it expresses a transgene. So you can design a probe to target the vector DNA on the promoter region. So now you know where your vector is in the tissues. And you can also say, okay, this is the transgene that's being expressed. And we can show for every one vector DNA, there's 3 copies of the transgene that's expressed. And you can do that in the same tissues. So yeah, okay, vector and transgene expression. And so here's an example of that. Again, these readouts, they're these little dots. So each dot is one RNA or DNA transcript. So here they showed that, for every one vector genome DNA, they had like 4 transgene being expressed. Okay. And then this is my last slide showing some examples. And I do this last because I think it's a cool slide and it shows you a lot of cool concepts with RNAscope. So this was used RNAscope was used to develop the COVID vaccine, but this is a paper published in Cell that used RNAscope during this time when we still didn't fully understand how COVID worked. I guess 2021 we probably did. But back, I don't know, you guys probably remember, we didn't know like where the COVID virus kind of hung out in the body or where it replicated. So on postmortem patient samples, researchers used RNAscope to detect the presence of the virus. So virus is a very small transcript, and you can design a probe to detect viruses. So here they used the normal antisense probe to detect the presence of the virus, which is these green puncta in olfactory epithelium. And then they thought, okay, so we know that it's in the olfactory epithelium, but where does it actually actively replicate? So they did what I talked about earlier, and they designed the inverse. They used a sense probe, which is only going to be specific to your actively replicating virus. And they identified that the COVID was replicating in olfactory epithelium. And then they went even further and they combined it with an antibody to identify which cell types, sub-cell types within the tissue COVID was being replicated within. And then finally, they wanted to look at these different variants. So this, you remember, there's a bunch of variants now, but there's the wild type and then the alpha variant, and using a BaseScope probe. So this is a very small probe. They were able to target just the region that is different between those 2 variants. So there's a lot of homology between the 2 variants, but there's small regions of difference. And if you can target that specific region, then you can identify the different variants. Any questions? Okay. All right, so now I'm just going to talk a little bit about this new protease-free thing that I talked about. I'll go over multiomic, and then I'll hand it off to Kelly. So in the past, we always used a protease to permeabilize tissue because it's very effective. It removes the protein, opens the extracellular membrane so your probes can enter your tissue. However, this caused a problem because a lot of researchers want to do RNAscope and then they want to do a protein stain. And as you know, protease degrades proteins. So in order to fix this issue, we came out with this protease-free reagent. This reagent is not enzymatic, so there's no protease or enzyme in there. They don't tell me what it is, but if you shake it, it bubbles, so you can probably figure it out. It's a permeabilization reagent that doesn't damage your tissues. So essentially, everything else in the workflow is the exact same except for this one reagent. And I will show you some images now of kind of comparing the protease workflow versus the protease-free. If you're already doing RNAscope and it works well, I would just stick with the protease if you don't care about the antibody. But if you'd like to combine, then I would test this new workflow. So here you can see the bottom is your normal protease workflow. So you have some nice-ish expression on the left, but your antibody, let's say you want to combine it with PD1 and CD8, the protease pretty much degrades your antibody signal. However, when you use this pre-treat pro, you still have really nice-ish signal. So we have shown that pre-treat pro, it still provides the same signal for the RNAscope portion, However, it doesn't damage your antibody. So now you can combine these things together, and then there's a nice overlay image. And then I got one more image. So this is new-end. So this is neurons in the brain. Again, protease destroys your new-end signal. However, if you replace that protease with we call it pre-treat probe. That's the name they come up with. So it's this protease-free, and it's called pre-treat probe. And it really preserves your new-end signal. All right. So now just going to go over this newest assay that we offer. It's called the RNAscope multi-omic assay. So the idea here, multi-omics, so there's two omics, protein, RNA. What we did is, remember how I was talking about how there are these specific channels that are defined by this sequence here, like this is your C1 probe, C2 probe. So we took the top portion of our RNAscope probes, the hat of the probe, and we directly conjugated them to an antibody. So now you don't even need to use a secondary antibody. You would just do the same amplification process with the RNA, like you would with normal RNAscope. It amplifies both the RNA and the protein signal, and then you can add a fluorophore using our typical method, which is HRP, deposits HRP, like pteramide signal amplification fluorophores, TSA dyes. We also took this top portion and we stuck it directly to a conjugated secondary. So if you want to use your own primary antibody, like you have a nice mouse IBA1 that you like, you can use our secondary antibodies and you can still have the same kind of amplification, so you get more signal. So like, for example, if you're trying to combine two different rabbit antibodies in a workflow, you'll have cross detection with your secondaries. So you either have to strip them in between, and that's kind of a pain in the butt, and also it can damage your tissues, or you have to find a new antibody that's not rabbit. But now the good thing about this assay is that you could just use like a C3 new-end or a C4 IBA1. It doesn't matter that they were both taken from rabbit because you're not using a secondary anymore. Does that make sense? Okay. And here's some nice pictures of results. This is a assay with 2 proteins and 4 RNAs. So you got CD163 and CD68, and then a bunch of these nice interleukins, cytokine kind of stuff, TNF alpha, IL-10. And of course, you can turn these channels on and off for quantification if there's too much signal obscuring one another. And finally, this is another nice picture of the mouse hippocampus. So these are all neuronal genes. You've got, your microglia, your astrocytes, your excitatory cells, oligodendrocyte, oops. And yeah, these are just 6 RNA targets altogether. And yeah, so that's something that we offer, and that's available on the LicaBond. Speaking of LicaBond, allow me to introduce Callie right now. Okay. Any questions? All right, thank you.
Thank you very, very much. And I just don't want to trip on cords. All right, so I'm really excited to talk about how we use ACD's multi-omic 6-plex assay. So I'm going to give you a demonstration of what we've done in-house, but most importantly, I want to talk about Halo AI. And so that's the platform that we use for the image analysis. So I think this is a great opportunity for us to convey the technology that we have down at HistoSpring that could be useful for many of your imaging needs. So Halo AI is a trained by example deep learning image analysis engine. So it's built for digital pathology. And so you don't need to write any code. Instead, you teach the system by drawing annotations of your slides, and you define classes that you care about, whether it's the epithelial tissue versus the stromal tissue, there's multiple examples that could be used. So the Halo AI learns visual features of those classes, and it applies them across the entire slide or all of the slides within your experiment. So it starts with annotation where you define the tissue regions and the cell types, and that careful annotation becomes the training that guides the AI models. And it uses real-time tuning so that you can make adjustments to the algorithms while it's actually working. And you can do this by adding additional annotations or training examples. And so from there, you can use the software to perform automated phenotyping, which classifies cells consistently and reproducibly. And it provides visual biomarker cues. And so you can literally watch expression levels change cell by cell and region by region. And it uses color-coded masks in order to distinguish between these defined areas and cell types. And then there's density heat maps you can use to visualize cellular hotspots and neighboring interactions. And finally, what is probably the most useful aspect of this software is that the system outputs quantitative data sets. And so these are all, things like cell counts, density, intensity metrics. And so these are all ready for downstream statistics. So it's powerful because it keeps the science visual and interactive, yet completely data-driven. So now that you've seen how Halo, our HistoSpring, combines the automation with our like a bond and Halo AI for quantitative analysis, I'm going to show you the example of how we quantified our results using ACD's multi-omic LS assay kit. Okay, so where we started. HistoSpring has access to over 650 tissues from patients that have undergone a breast surgery following a cancer diagnosis. So we selected a breast tumor and we performed an H&E automated at our site. And then I took the slide and I scanned it. And we have an Apereo CS2 scanner, which scans bright field images. And so once I did the scan, I was able to see the entire slide, and you can use freeware either from Leica, which is the ImageScope technology or ImageScope viewing system, or you can use QPath if you're a Mac user. So when you do that, I don't have the example here, but you can actually scroll all the way into the tissue up to the magnification at which you scanned. So it's a great way to be able to visualize the entire slide without having to sit at a microscope. And so here we can use this guy. So that's our H&E. And then we used the multi-omic kit. And here I'm going to demonstrate an example of a 5-plex. As Blake said, we can go up to 6. So this fluorescently stained slide was scanned by the UMass Microscopy Core. Okay, so here in this panel, what we decided to look at. So we looked at our immune markers, which Hank talked about before, but FOXP3, CD4, and CD8. We looked at an epithelial marker, which is pan-CK. And finally, for our RNA probe, we looked at RRM2. RRM2 is important for DNA synthesis and repair. And in fact, RRM2 expression is downregulated in senescent cells. So this multiplex image gives us an overview of the breast tumor microenvironment. And it reveals how the immune infiltration and epithelial integrity and how DNA synthesis coexist within the same piece of tissue. All right, this is a high resolution image. And this image allows us to move beyond just counting cells. It helps us understand how there's spatial relationships. And so using spectral scanning and digital overlay, we can examine how those interactions happen at a single cell resolution. Okay, so this is what we see, and this is what we're all used to seeing in papers, right? All of our beautiful multiplex images. But now I want to discuss in detail how we use the Halo AI to extract quantifiable and numerical data from this histological image. Okay, so the first thing you do is you upload your slide into the software. All right, and that's your entire scan slide. And so once you do that, you need to start training the software to output what you're interested in. And so the first thing you need to do is you need to create a classifier. So this is Halo's MiniNet classifier. It's a deep learning segmentation engine, and it's trained specifically on this breast tissue. So this is how I did the training. So here you can see this yellow rectangle. I'm trained this is my glass. So yes, I am including stroma and epithelium. So, but you have to think of it as a layer. So my least important aspect is my glass. And then in red, it's a little bit harder to see, but in red, I'm training the software where all of my stroma is. And then within these stromal regions, I have to tell it where the epithelium is. And here I talked about that mask. And so what you do is once you start the training, you see a real-time tuning window. So you can see the machine learning actually adjust the algorithms in order to accurately depict the tissue that you're trying to define. So this example of a mask is there's a little bit of transparency. So I can visually see that I'm happy with the software's ability to delineate between those two tissue types. And here is just a full mask fusion version of what I'm able to see. Okay, and another newer aspect of Halo is their probability mapping. So these displayed masks right here, they represent a confidence overlay. So the AI assigns probability to each pixel or cell, indicating how certain it is that the region belongs to a specific class, say the epithelium. So red indicates that it's very certain that, you know, the tissue is what you trained it to be. Where it's blue, it's very certain that it is not the tissue that you trained it to be. So these other areas in here, these ambiguous regions, you can actually, in real time, offer more training in order to refine the algorithm. So it's a quality control tool that builds trust in the AI's prediction. Okay, so let's put this all together. All right, so now that we looked at the image slide, I want to walk you through how I turned those that multiplex image into the quantifiable data that I've been talking about. So for this analysis, I used two modules. I used their high-plex FL module, okay, to look at the IHC or the protein stains. And then I used their FISH, which is fluorescent in situ hybridization, to measure the RNA expression. So, okay, so the first step, what I need to do, so all the blue, as we all probably know, are the nuclei. So the first step in the software is to define the nuclear detection. So I can change a whole bunch of different parameters. I can change the minimum nuclear intensity, nuclear size, nuclear segmentation, nuclear aggressiveness, and the minimal nuclear roundness. Okay, so luckily Halo comes with a pre-trained AI or a pre-trained fluorescent nuclear detection. So you can just upload that classifier into your software and you can make sure that, okay, yep, these are nuclei. Because sometimes it doesn't pick up the nuclei that are there. So you can just make a little bit of adjustments with those settings. And the same thing goes for the membrane and the cytoplasmic detection. So different parameters that you can change. So here in gray is how I define my cytoplasm. Is you can change the membrane segmentation type, and then you can also introduce one of Halo's pre-trained membrane segmentation classifiers. So these parameters ensure that each cell's boundaries are accurately recognized, and that by distinguishing the nuclear, cytoplasmic, and the membrane compartments. So the next step is to train the different locations for each marker within the multiplexed image. So let's start with here. We have our CD8 and our CD4. Okay, so those are cytoplasmic proteins. So what I did was for each one of these IHC markers, you have 3 different things that you can modify in order to make sure you're accurately picking up the stain that you're interested in. So for here, I adjusted the cytoplasmic positive threshold, okay? For FOXP3, right here, this is a nuclear transcription factor. So the only thing that I needed to modify in that setting was the nuclear positive threshold. And lastly, pan-CK down here, that's our membrane protein. Okay, so, so basically I adjusted the settings for the membrane positive threshold. Okay, so then up here is our RRM2. Okay, this one is a little bit more complicated. That's why they have a separate module for it. Okay, so this is our fish probe. So as Blake had mentioned, nicely with the chromogenic stains that he showed, is that each RNA transcript is basically expressed as a fluorescent dot within the nucleus. So the software, HALO, quantifies both the number and intensity of those dots per cell, allowing true single cell RNA measurements within the intact tissue. So the parameters that you can adjust within the software to make sure that the signal you're getting is true and not background include contrast threshold, spot size, signal minimum intensity, as well as copy intensity. Okay, now the last step, remember I did my classifier with MiniNet to distinguish between the stroma and the epithelium. What you can do is you can import that classifier into your analysis tab. Okay, so this way all of your metrics, cell counts, densities, and transcript numbers are all automatically reported in total, but you can also have them reported within each defined compartment. Okay, so what this gives us is an integrated data set for protein expression, RNA copy number, spatial localization, all lined up cell by cell and region by region. So it's a powerful way to move from multiplex imaging to quantitative, reproducible, and biologically meaningful results. So after I ran the analysis, I extracted the numerical quantitative data. So next what I'm going to do is I'm going to break down this data by showing you a couple tables that depict both our IHC as well as our fish results. Okay, so what I'm going to show you here, so as I mentioned just in talking, you can get all of your total data. Your total data is exported, but what I'm showing you is how I've been able to use my MiniNet classifier to get data that's expressed just within each component. So we've got our epithelium and our stroma. So we've got total positive cells within each compartment, the number of cells per micron, right? Because I have the area of the entire tissue based on my classifier. I've got the percent positive cells, but also because I've defined those other regions, my nucleus, my cytoplasm, and my membrane, I can get the percent of positive cells within each one of those compartments. So you can see here, so our nuclear cells, right? FOXP3 is our transcription factor. You don't have any expression. The only place that you have positivity, right, is within the nucleus. Percent positive cytoplasmic. We don't have anything for FOXP3 or PanCK, but we do have outputs for our percent positive cytoplasm. And then for our membrane, same thing goes for the PanCK. And so not only do you get positive or percent positive cells, you can also get intensity readings from each one of those readings or each one of those cellular compartments. Okay, so now let's talk about the RNA side of the analysis where we're looking specifically at the RRM2 probe. And so different parameters that we captured here are, and again, per region is the copy number, the area of the expression, the average copies per cell, the average copies per cell per area, and the cellular intensity, and the copies per area. And lastly, what you can get is you can get what's called an H-score. So an H-score is a semi-quantitative measure of expression intensity across all of your analyzed cells. But what's broken down here is you have a 1+ down to a 4+, okay? So that's basically the percent copies per cell. So here, a 1+ is indicated by 1 copy per cell. A 2+ is indicated by 4 copies per cell. A 3+ is 10 copies per cell. And then if you have a 4+ score, that's 16 or more copies per cell. And so it's not just by adding the numbers, but there's a little equation. It's pretty simple. But using those numbers, you can generate your average intensity score. All right, let's recap the big picture. So first, spatial biology is reshaping how we're studying disease, right? Like look at the example of COVID. It gives us the ability to see molecular and structural context simultaneously. And RNAscope remains the gold standard for in situ RNA detection. Okay, so it's been cited in over 13,000 peer-reviewed publications, and that's for a reason. Third, pairing RNAscope with HistoSpring's histology, as well as image analysis capabilities, transforms qualitative observations into quantitative outcomes. And then finally, our combined expertise allows us to deliver high-quality, reproducible data. And that accelerates translational and applied research. So together, we're not just visualizing biology, we're quantifying it. Okay, so I'm gonna bring Blake up here one more time, and he's gonna talk about getting started with RNAscope and what a typical workflow process looks like when you pair with HistoSpring. Next. And advanced slide, that's all.
All right. Yeah, so, I'll pause it down. Yeah, so like if you ever have an experiment that you want to use RNAscope for, obviously it would probably involve RNA. So essentially this is the workflow that I would do when trying to think about experiment is first think about what you'd like to look at. So are you looking at just RNA? Are you looking at RNA and protein? What would you prefer? A chromogenic readout, fluorescent readout? You don't have to know all of this stuff. But just kind of start thinking about it. So yeah, step one, RNA or RNA and protein, because then we would go with the protease-free reagent. So there's kind of some nuance there. Yeah, chromogenic fluorescent. For example, a lot of pathologists like chromogenic assays because you have that counter stain, so you can see the morphology of the tissues and you can kind of localize signal that way. Or let's say you want to do 4 targets, you don't want to do chromogenic because it'll just look like a blob. So then you would do fluorescent. And then finally, the very important thing is tissue processing. So your tissues do need to be fixed correctly or else there's not going to be any RNA to target. So just know if you're going to do frozen tissues or if you have paraffin embedded tissues. And then once you have all that information, you can meet with us essentially to plan your experiment. So we've got Hank right here. He is your account executive. So he knows a lot about RNAscope and he can also loop myself or a different field application scientist in to kind of just meet with you and discuss your project. Because I've been doing this for a long time and I feel like just having a 30-minute meeting and planning out the experiment saves so much trouble in the long run versus when people try to figure it out themselves, because it's dense. There's a lot of weird stuff that can kind of go wrong. So when you meet with us, we make sure that that doesn't happen. And then I just press next. So is this you or I'll do it. Oh yeah, no, this is me. Yep. Okay. So again, I said earlier that we can design any probe to target any gene in any species. So if you do need a probe designed, that you would just submit the sequence to us. There's a form online, takes 24 hours to get a response. And then if you actually want to purchase that, the probe will arrive at your lab in 2 weeks. So it takes 2 weeks to synthesize new probes. But if you're just looking at a normal gene, we probably already have it designed. We have probes, 50,000 probes designed. So then we already have that ready. It'd probably ship out the same week. So getting the kits and probes, that's about 1 week. Sample prep and drop off. So this is going to be for HistoSpring. Do you do the sample prep? Or they drop them off?
Yeah, they would drop it off.
Okay. We can
just
share this one. Yeah.
Yeah. And then, yeah, so after we get the samples, then we're gonna run the staining on the bond. Sometimes if it's a newer assay and it's something that you've talked to Hank and Blake about, Blake or another field application specialist will come to our lab and help program our bond in order to be specific for the assay. Or maybe we're just rerunning something and we already have a predefined assay, but that's something that we all work together on. And then, yes, after that, then we go to imaging. So then if it's a fluorescent image, it would be submitted to the UMass Microscopy Core. And they use a tissue gnostics microscope in order to they can get up to a six dye slide and then plus DAPI on top of that. And then that would come back to me, and then I would be involved in doing the halo analysis. That could be really quick, or if you have something complicated, it could take longer. The biggest thing is doing all of those parameter threshold adjustments within the software, and that's something that I would work with you on in order to make sure I'm capturing the actual information that you want to be seen.
Awesome. So does anybody have any questions about RNAscope or HistoSpring or really anything? We'll also hang around afterwards if you want to talk about something.
Cost.
Cost.
4.
That's you, right?
So on the reagent side, it'd be, depending on the complexity, like 50 to 250 per slide.
Depends how many probes you have, which assay exactly, yeah. Yes,
I have two questions, for you. You said you could use either fixed tissue formalin fixed or fresh, which do you prefer?
yeah, good question. I feel like fixing the tissues is safer because depending, you can let them sit for longer in your freezer. But theoretically, fresh frozen might have the highest quality RNA because you take the tissue and you freeze it right away. So then, yeah, versus take the tissue, fix it in PFA for 24 hours. So it goes into, there's some time where it's just kind of getting the PFA in the tissue and the RNA has a chance to degrade there. I would honestly say that they are both totally fine and you should stick to whatever your lab has like a protocol for.
Yeah, and I'll bring up the example that I showed is a formalin fixed paraffin embedded sample. So we still got great detection.
Thanks.
Yeah.
The other question was for Kelly. Kelly, can you use the Halo software with GAB stained tissue or?
Yeah, absolutely. So here's a quick little example of, being able to detect, they have a predefined nuclear segmentation for bright field, and you can also mask the cells according to different intensities. So for example, Ki67 is a great sort of indication of what we can do here, and because a lot of times people want to report % positive cells. So I don't know who's done this, but I've done it plenty of times as I sit in front of a computer with a clicker and I count every single epithelial cell, and then I count all of the brown cells, right, in order to get a % positive. And I do that also, like I've taken pictures of each of like maybe 5 different sections of that tissue. What's great here is if you have a bright field scan, you have the entire tissue. And then second of all, the AI can delineate what just is epithelium. So you have the AI to define your epithelium. And then you can also, so these are different colors, but what I'm not thresholding, I can just get the % positive cells. And that's very easy. You can take it a little bit further if you want to. If you want to go to the next slide. Okay, so here's an example of something that's harder if you were just trying to get quantifiable data from this image. So what you can actually do is you can go in and you can define the nucleus and you can define the cytoplasm and you can threshold within those regions in order to get different intensities. So again, these are just 2 different masks. This is my transparent mask and this is sort of my virtual mask. And so what I can get is I can get weak, moderate, strong, and negative nuclei. Okay, and I also can get a percentage of those same values. And you can do the same thing. So that's looking at both cytoplasm. So I can do this also, most importantly, I can do this for the nucleus, and then I can also get the same data separately from the cytoplasm. And lastly, what you can do is you can do the same thing for a membrane stain. Okay, beta-catenin, I think, is this example where it's a membrane stain, but it's also nuclear. So it's really important to know whether it's nuclear or membrane based on, the biology of what's going on. So you can actually get those same sort of informations in the nucleus and the membrane in terms of the weak, moderate, strong, and negative.
Real quick, so if we want access at UMass to this Halo AI, it would be through contract with HistoSpring.
Yeah.
Okay. And so, but we could also do that if we did the manual RNAscope assays.
Yeah. So,
So you could do either, but you could either do, say, get something done on the Laker bond and analyze it here ourselves, or vice versa, do something here ourselves and analyze, okay.
Yeah.
And then for the Laker bond, what about, because you're a histology, like human histology core, what about using other species tissue? Is that?
100%. Yeah,
Okay.
yeah, because we're not a clinical lab. That's, we are an academic lab, and we've seen a lot of different species come through. And then you said for the RNAscope, any species.
Yeah, I mean, people
Yeah.
even do plants.
Yeah.
Yeah.
We've done a couple of species, but yeah.
Just maybe for Kelly also, so once you do the Halo and the training, then the first time you do it for whatever I might be interested, might take some time and be longer, and then it.
That's exactly it. So this would be sort of a fee for service, it's a labor intensive thing. So in the beginning, defining all the parameters is most time consuming, especially if you want to make sure that they're, you know, the researcher likes them. But then once it's been trained and you give me another set of slides,
So the cost goes down?
significantly.
Okay.
Yeah, because all I have to do is upload them into the software, and then, because I save everything, right? So all I have to do is upload all the analysis, you know, module that I've already sort of trained, and then click analyze.
I have a question, I think for Blake about the RNAscope stuff. You said at the beginning that sometimes the puncta are like very dense, and on that slide, it kind of looked like they were like larger. How do you know that they're not obscuring another signal when they're, that, it was like a very early on.
Yeah, I know what
slide
you
that had.
mean. This might be a halo thing, but essentially, so yeah, if you have high expression, you have clustering. Then you have a bunch of these little puncta, they all are overlapping, then it looks like kind of more of like a blob.
Yeah.
So that was I know the slide you're talking about. And
I don't have a blob here.
yeah, so what people normally do is you'd use some sort, it doesn't have to be Halo, but it would just be some sort of software. You define the average dot size. So you'll say the average puncta is this big, and then you circle the ROI, and then the software makes an estimation, and you would say, okay, because there's so much signal here, we're assuming that there's 20 puncta in this region. So you wouldn't be able to cut, you know, it's more of a, yeah, you're taking an estimate. Is that right?
Yeah,
Is that what you do?
Yeah,
Okay.
so I think what I can go here is I can show you, that's kind of a blown up version, but I have a little bit more information that I didn't talk about when I was giving the talk, but the things that you can change, like I said, the contrast threshold, the spot size, the signal minimum intensity, and the copy intensity help this right there. So you, like, the contrast threshold can tell you the difference between signal, if it's signal itself and background. The spot size, you can adjust what's being detected, right? So that there's big blobs, but you can put a minimum and a maximum size, you know, for the software. And so there's a signal minimum intensity. So if you lower this, you can lower this if you're missing out on some of the weaker signals, but you can blow it up, you know, if you're picking up some stuff that you don't want to be measuring. And then the copy intensity, so that determines the number of copies within a cluster. So for instance, I think the example that when I was working with the folks that were training me on Halo AI. So I think I have this copied intensity like at a 30, right? But if something comes up as a 40, it's actually going to be detected as 2 signals. So you can change the parameters in the software for that specific.
Yeah, I'm just thinking about the flow. If I had 20 slides and I've done them manually, and then I'm trying to use Halo to analyze, I have a little more variation from slide to slide. Do you think that would cause much difficulty for Halo to have the same settings when the slides were done at different times?
You could modify your, so you're saying you did an experiment, you had 20 slides and I've trained Halo, you know, according to the parameters and the specifications to be happy. And then you do 20 more, but there's variability and you don't want it to be analyzed the same way as your previous experiment. But we could make some tweaks in order to make sure we're capturing, you know, you can make sure the mask is the same, you know.
So I'm just thinking that the bond automated thing now that you can multiplex, that would take out a lot of that variation, or have you?
There shouldn't be that much variability. I know you're saying, yeah, if you do it by hand, but it's relatively like stable, I guess. Like the incubations, if it's 5 minutes off, shouldn't be that much variability. But
Yeah, I
Yeah.
agree.
the like a bond, yes, does reduce variability because there's no human doing it. So it's just runs, it's a robot. So then there's way less variability.
How long does it take to do a 5?
So this 5-plex took 20 hours. Yeah. So I set it up on a Tuesday and I got my data on Thursday. I actually got the slide and then I brought it to you guys.
Yeah, it runs overnight. Yeah. So.
And you can delay, I mean, as a researcher, you can delay the start, so it starts at a certain time so that when I come in and it's off or whatnot, so. But yeah, it's a long assay. Christian, or my field of application specialist or scientist warned me.
Yeah.
Do it earlier than me.
Yeah. Any last questions? All right, well, thank you guys for coming.